The purpose of this project was to test the tolerance range of pH for Chlamydomonas, along with how this affects its lipid production, which will correlate to its ability to become biofuel.
Chlamydomonas is a type of biflagellate single-cell green algae. It's most commonly found living in soil, ponds, and ditches, and some species of Chlamydomonas are able to live in snowy areas. According to a study done by Messerly and others, this algae's optimal growth is usually in a pH of 5.5 to 8.5, but may grow outside of that as well.
Because of this, we chose to study its growths at pHs 3 through 10 and to test its tolerance. From the previous research we've done, we've then hypothesized that the algae would have the highest growth rates between pHs 5 and 8, with less growth in the lower and higher pHs.
First off, we adjusted the pH of the pre-made tap medium by using hydrochloric acid or sodium hydroxide. To ensure we had the correct pH, we used a pH meter. We then added 2 milliliters of the adjusted tap medium into spectrometer tubes, and this was done in replicates of 3. We pasteurized them to kill any possible contaminants. We then added 2 milliliters of the pre-ordered Chlamydomonas, and we exposed them to light for 16 hours and then no light for 8 hours. We inverted the tubes to suspend the algae throughout the medium and measured the growth by absorbance at 600 nanometers using a spectrometer.
As growth stabilized, we centrifuged the tubes to form a pellet of algae to isolate the cells for the lipid assay. We discarded the tap medium from the tubes to then isolate the pellets. Next, we froze the tubes at negative 80 degrees Celsius until every sample's growth had stabilized. We rehydrated the samples using sterile water and moved them to microcentrifuge tubes to obtain their wet weight. We centrifuged the tubes and removed the liquid and freeze-dried the pellets. We stored the pellets at negative 80 degrees Celsius until we were ready to take the dry weight. We then measured the tubes to be able to calculate the dry weight of the pellets.
Following the procedure laid out by Chang and fellow researchers, we added 100 microliters of chloroform and methanol mixture to the dry pellets. We then vortexed the tubes to ensure that the cells were blended within the mixture. We centrifuged the tubes to remove all cell components except the lipids.
We moved 50 microliters of the mixture to microtiter plates. We allowed all of the liquid to evaporate from the wells and prepared a standard plate using an oil and chloroform mixture. We added 100 microliters of sulfuric acid to each well. We incubated these plates at 90 degrees Celsius for 20 minutes. We took initial absorbance readings at 540 nanometers with a microplate reader. We added 50 microliters of vanillin phosphoric acid and let them sit for 10 minutes and then took readings again.
The results support our hypothesis that growth was highest between the pHs of 5.5 and 8.5, as well as lower growth at the extremities.
This graph further supports our hypothesis, though technical error gave a negative value for pH 5.
This graph shows the correlation between lipid production and environmental pH.
Research done by Kostrov and Associates found that when the pH is out of optimum range, hydrogen production decreases, which hinders metabolic processes. The lipid data from our experiment suggests that the pH can impact the algae's ability to produce lipids. This can then affect the ability to produce Krebs cycle intermediates from lipids in times of glucose deprivation. In pHs that impact lipid production levels, this could potentially hinder the algae's ability to survive in certain habitats, potentially decreasing its survival rate.
As shown by Bell, there's a strong correlation between environmental conditions, growth rate, and genotypic expression. This is also supported by our data. This becomes important to determine the algae's chances of survival due to the increasing acidity in their environments because of current levels of pollution. In conclusion, environmental pH has a profound impact on the algae's ability to grow and thus affects its lipid production and survival rates.